ROV setting down.
Aaron, let's push it.
Messing with stuff would
hold that.
Replumb it a bit.
Right, right.
From the Endeavour event field, I still think that's one of the like craziest facts that I walked away from that Rennie with those expeditions we've done up there is that there are tidal variations in the venting.
Yeah.
I like that even at the bottom of the ocean, the amount of water you could think about stacked up on top of them as that changes with tidal cycles, that changes the flow rates.
Yeah,
Like Earth is a big squeeze toy, you know, and like when is more water squeezing on it or releasing that pressure?
Right.
We think we see that something similar to that with methane seeps as well. They're kind of ephemeral and change often. And you can see that in the communities down there too.
How much more zoom do we have, by the way? Just curious.
Video pushing all the way.
Oh, it is? Okay. Can you go down?
Now we're in all the way.
Now we are, okay.
Yeah.
You can come back out a little bit.
Sure.
And in the lower left right now, there's that octopus with eggs visible. Can we focus in on that or zoom in on that?
Sure.
I want to see, it looks like it might be getting senescent, so the eggs, I wonder if are have developed embryos inside.
That's a nice spot.
Yeah.
Wow.
So it looks like an older octopus, so yeah, that's, the eggs are way more developed, so you can see the eye spots there.
Wow. What is the clue that they're senescent? Is it the marbling?
The marbling, yeah.
Okay, and what does senescent mean for all of our viewers?
So these octopuses, we don't know how long they brood.
Right over there.
I guess probably a year to two, maybe even more.
Wide on Argus.
But when the mothers sit down and lay their eggs, that's it. They're done in the sense of they don't eat anymore. This is their mission in life, is to lay their eggs,
Back in a little bit.
take care of them, make sure they develop, and then hatch, and then their body essentially wastes away.
Unless
all
you're
their
using
energy
a bubble on
stores
the end of
are
the arm,
used up, and they
I don't have any camera on
essentially deteriorate and pass away. But this is this whole cycle of life with these guys.
And the deterioration is senescence.
And the deterioration is senescence, exactly.
Yep. All right, we don't seem like we have any kind of rapid temperature climb here, so I'm going to poke to a different spot.
Yeah, go ahead and check a couple more spots.
All right.
Great.
Thank you, Aaron. You can hold that. I'm going to go by where those shrimp are.
Chad, what do we know about what type of octopus these are?
We have a colleague of ours, Janet Voigt, who has worked. She's a cephalopod expert.
Was where all that shimmer is right there.
So we were able to send her a specimen, and she's fairly confident, but it's not a positive identification at this point, that it's Mus octopus robustus. So it's M-U-U-S octopus.
In the center screen.
Yeah,
robustus. There's 13 or 14
species
species in this genus.
This one's at least increasing
and
in
this genus is the only one of the octopus that brood in this crazy upside down position.
Awesome.
All right, that
And I
temperature
wonder
looks
if
like
that
it peaked.
brooding
Didn't come up
position
very
is a
much
function
anyway.
of living in rocks. So they have their defenses outward and the rest of the body and the eggs sucked down into the holes.
Yeah, that's an interesting idea. I sort of thought, you know, if you're aerating, you'd want all your legs, your swirly parts down towards your eggs, but.
Yeah, their legs are really still.
I'm curious just where the temperature probe is on this being
It's all the way up right by that cutout.
I mean, I'm curious where its position is while it's, if it's measuring, if it's too far away from, because we see just even centimeters away, we'll see a steep drop in the temperature.
Yeah, if the little schnozzle's too long.
Yeah.
Yeah. It might not be reading, it might be close to seawater by the time we get
Aaron, let's
maybe
come full wide.
if we can try 1 or 2 more spots, I'd love to try the other probe too.
So can you explain for the watchers what the term schnozzle
This one is
means?
increasing, so.
Oh, schnozzle is a very scientific term. We use it frequently here on Nautilus. Any instrument that has a tube, a
1 degree, 0.1
prong.
degree.
Yeah.
The slurp sampler, often.
I'll give it here just so
that
a little bit more.
Boy, now is that huge anemone there the same as those smaller ones?
As far as I know, yes.
That's amazing.
They have that warty column.
Yeah.
And the same kind of arrangement of the tentacles. I think it's just a larger, older individual.
Okay.
And I'm forgetting the species name, but this particular one is not unique to these vents. It's kind of cosmopolitan down here. But they are concentrated in these vents.
Interesting.
Nope, we're going up again, but still just like barely.
Jamie, what kind of temperature are you seeing? I can't actually pull up the computer up here.
We're at 2.4.
2.4.
Something we should comment on with here, because we've got the min and the max down here, but we don't actually have the readout. You have to look on the graph for that.
Yeah.
That was still coming up.
Video push in.
So the measurements of over 10 degrees,
right,
Chad,
if we're
where were
going
those
to
from?
10.
Those were from the vent,
Okay.
which we were just at from the previous shift. And I believe they got temperature as high as 9 today.
There's a bit more zoom if you need
but
it.
that seemed to be a place where it was essentially a column or chimney.
Right.
And the seeping water would be more concentrated, I guess, and less mixed with the bottom water.
Yep.
While obviously this place, it's going to be a lot of mixing with bottom water because of the complexity and the kind of the rock structure here.
Right. It's like coming out through a big diffuser.
Yeah, yeah, exactly. And with the other probe, which is similar to what we used in August, you know, you could move the probe, shove it into a crack, maybe another couple centimeters, and suddenly it would jump 3 or 4 degrees. So,
right.
Definitely a bit of art and luck to getting high temperatures
right.
here.
Definitely.
So there is art in science.
100%.
Yep. Just like there's intuition. Intuition, creativity, all those things go into doing this.
Yeah, the question keeps coming a couple times about how large these are. You can.
I found a spot.
We got a we got a rapid rise in temperature. We got up to 5 degrees right away.
Oh yeah.
Good job, Greg.
Good spot. So you can scale things yourself as long as those 2 green dots that are in the video feed on camera 1 or channel 1 on the website are in view. So it's on the arm right now, so a little tough, but the 2 dots are 10 centimeters apart, and you can use that just like a ruler, 10, 20, 30, 40.
I see a reflection off the arm onto the octopus below it.
Ideally, you have them hitting the same plane.
Right.
What's happening over on the right?
Yeah, we got a little bit of a tussle going on here.
Thunderdome.
So, yeah, I mean, we don't know much about the behavior of these mothers, but we have seen this kind of interaction before, and I wonder if it's, you know, this is my turf, this is where my eggs are going, or my eggs are.
You wonder if that's why they why they protect their eggs.
Be.
Now, those are some
tentacles
spent egg cases
faced
there. You
out.
see right between them?
Yeah.
So those are empty egg cases, so I don't know whose those belong to, but...
So you can better defend if your arms are out. Maybe these areas are hot commodity.
Slap down.
Let's come down 3 meters.
Okay.
Just because we're starting to get a little bit of tether tug.
Yeah, it's it did cross over 6 degrees Celsius now.
Yeah.
So yeah, let's see how far this rises. But I think this might be a really good spot to put one of the loggers in, and to potentially take.
I guess
look at all that shimmer to the right. Lots of water coming out.
Does that one have a weird leg?
Yeah, it looks like it's torn.
Yeah, it
It's
is.
missing an arm.
Mm-hmm.
Yep, it's been torn off.
We've seen this behavior in some others. We have an amazing highlight clip on Nautilus Live of 2 giant Pacific octopus sort of wrestling in this same configuration.
And it's almost like the one on the right is pulling the other arm in toward its mouth. Wow. Well, the one on the right
Let's
certainly
put
looks
bubble
bigger.
on the arm.
Mm-hmm.
Thanks.
Yeah, you're right. You're right about that, Dane. And so we've often seen the males.
That's how you know you're in the right
are
spot.
quite a bit
You're
larger
right
than the
behind.
females.
All right, so temperature is dropping, but we hit 6.
Can you
So
see
we hit
that
6.
specialized?
No,
Yeah.
it would be arm number 3, which would be on our left,
Okay.
sort of behind everything right now.
Right.
So, yeah, I think it would be very hard to see if we could find it. Now, the females, we've also noticed are just larger before they lay their eggs, and I think as they're brooding, they're consuming resources, wasting away, maybe not as swollen with seawater, so they tend to get a little smaller the longer they brood.
Okay.
But
Video, let's come full wide.
I mean, it could be a male. Trying to,
I know that you
I'll
just
be able to reach that other spot.
got rid of your embryos. It's time for us to meet.
Video, let's push in.
So Chad, we have a high school oceanography
let's
class tuning in right now, and they would like to know if there's anything special about the chemical content in the water,
Okay, let's remember that last spot. That was
is the
6.
increased temperature only right here in this area? Could you give them a little introduction?
Sure. So what we think this is, that's a mouthful, but it's a low temperature ridge flank hydrothermal system. And if it is that, which we do think it is, and I'm not a geologist, it would only be the 3rd such system discovered in the world, although it's presumed there are millions of these places. It's just they're often in places we don't visit. So essentially there's a large extinct volcano just about 8 kilometers away from us. We're on the flanks of it, the very base of it. So seawater is probably traveling through the porous basalt volcanic rock of the seamount and then traveling underneath probably 200 or 400 meters of sediment and through the top layer of basalt underneath everything, getting slightly warmed up by the Earth's heat and then coming out in weak points in the Earth's surface, which would be right here, kind of where there's a fissure or crack or fault or break in the rock. So it's just escaping via pressure, hydrostatic pressure, but it's been slightly warmed. So even though it still is in the low 50s, it's still cold ocean temperatures, it's near freezing down here, so it's relatively very warm to these octopuses. So the mineral content sometimes changes when it goes underneath the Earth's crust, so that's what we're going to be doing, is collecting some seawater to do some chemistry analysis. And also typically a lot of oxygen is stripped out of the water too, which is kind of the paradox, because these octopus eggs need to be oxygenated, and we've already seen today in the previous place oxygen levels about 50% of ambient, and in another place near the Dorado outcrop, which is really the only other place in the world they've discovered large groups of octopuses, and it was only 100 there. They never found any viable embryo development, and no hatchlings were ever observed, and they attributed it to the low oxygen concentrations. Here, they are very successful. We just saw some developing embryos. Earlier on the previous shift, they saw over a dozen hatch. On previous cruises, we've seen several hatch. So this might be an actual nursery, and so if the oxygen levels really are very low, they're clearly adapted to it or have no problem developing their young here.
Awesome, such a good explanation.
Chad, what about sulfides in the water? Are you measuring those?
That is part of the analysis. I don't know what levels they are expected to be at. So yeah, we've hit, what, 7.5 degrees here?
Yep.
Yeah, so we're using that probe. We're checking out different areas where we see a lot of shimmering and trying to see what the max temperature we can record in this area is, so we can deploy some sensors that'll stick around.
So the highest we've gotten here at the spa is 9.7, but that was with like your Hercules probe.
Right.
So after this
it's come down a little bit
reading
more
stabilizes, I'd love to get the other probe out and just for comparison's sake, stick it in the same area and then probably deploy a logger here.
That didn't pull tight.
If the last,
yeah, we don't want that female to latch on too much. There you go.
Oh, we hit over 7.5.
Oh yeah, looks like almost 8 there. This will be a really good spot.
Yeah, you win.
I think it's a marble perch or something.
Thanks for the questions. Go Wolverines. Keep sending them in. Oh, here's an interesting question, Chad. Can this mini octopus actually be altering the chemical composition of the water around it? Would you see a dip in the oxygen from so many animals being in the same area, or a change in any of the other chemicals?
Immediately adjacent to the animals, there probably is a slight change, but their respiration rate is relatively low, especially here in the deep sea. Everything kind of metabolizes and respires much slower than warmer, shallower waters. And there's just so much volume of bottom seawater kind of in a current sloshing over this whole area. I think mixing whatever may have been reduced right away. So I don't think there's any appreciable effect to any other organisms in terms of them consuming the oxygen, especially considering it's already relatively low when it's coming out of the seafloor.
But in terms of other chemicals like metabolic products, maybe talk about what they found at off Costa Rica in terms of these guys sitting in one position and not moving, feeding for a long time. They really slow down their metabolism. So,
yeah, they're not quite in a stasis, but I mean, they do have to exist for probably at least a couple of years, and they're not consuming anything. They're not taking in any energy.
It's wild. And I think a lot of viewers would be familiar with this idea that bears get really big and round and then they go hibernate. But I mean, the more we learn about bears, it's not like they go to sleep for like half of a year. Like they wake up and they might eat more food during the winter. I happen to have a bear expert sitting right next to me, so I was comparing it with the idea that most people might be more familiar with like bears hibernating, right? Eat a bunch of food and then rest, but then bears wake up and they eat more food. Not a perfect analog to what's happening here.
Someone else described it actually like salmon, where they spawn and they stop eating once they hit fresh water. And then they protect their redds or their nests until they die.
Yeah, that's much better.
One of the scientists described it as that earlier.
We're warming up, getting our analogies all warmed up. Start of a cruise. And why do they not eat for years? They are defending eggs, brooding over eggs. It's just a really unique aspect of the life cycle of octopus, is that this is sort of the pattern they have developed. So they are oxygenating their eggs, defending them from predators, so they don't have the opportunity to, get up, leave, go find some food, forage, and then come back. That's not the behavior.
So is it just deep sea octopus that take this long to brood then?
Yeah, there's not been a whole lot of study on them. So there was one species, a different genus in Monterey Bay that was visited again and again and again by Monterey Bay Aquarium Research Institute. But it was a solitary mother they were able to mark it. So they knew it wasn't a different mother every time they visited it. And it took 54 months till her eggs hatched. So that's 4.5 years. So it could be an outlier, that could be the maximum, but that could be standard. Everything does happen a lot more slowly here in the deep sea. So we have not been able to mark these particular octopus. Of course, we discovered this just a year ago, but in the future we would like to come up with a system of marking mothers and understanding how long they're here. But it's presumed it's probably going to be a couple of years at least.
With that one that you were studying, did she do the same thing where she didn't eat and then?
Yes.
Basically. So
yeah, once they lay their eggs, they're done.
That's crazy.
Wow.
Can we take a look at the octopus in the lower left? I think we have also developed embryos down there.
Chad, do we have an idea of how old these octopus are until they, before they lay their eggs? Are we talking a couple of years old, a couple decades old?
Yeah, probably not a couple decades, but most likely at least several years. They're cousins in the shallow waters, like giant Pacific octopus, even though they're so much larger.
You can see eye spots when the mantle moves.
They only live 2 to 3 years.
Wow.
And they only brood for 3 months. But again, that's the metabolism, their life cycle is much faster than these guys here. So, but again, we don't know how old they are. And now that we've seen hatchlings for the first time earlier this year, and a lot more today, the one thing we haven't seen is the intermediate life stages. We haven't seen a teenager, an octopus that's halfway between in size. We do see them kind of like Charlotte's Web. They just kind of disappear off into the water column, and we don't know where they go. We don't know how they grow up and where they grow up, and if some come back here or they reseed the population miles away. We don't know the answers to any of these questions.
Yeah, that's good.
What about general octopus biology? I mean, aren't they like the most intelligent invertebrate?
Before you get into
That's what I've been told.
Yeah.
Chad,
By an
we have
octopus.
hit 7.5 twice here.
Yes.
So do we want to
can we
use this as our...
Yeah, can we use the Herc probe?
Yep.
Let's do that, and then, we'll put a logger here, but I want to do a comparison between this probe and the Herc probe, just kind of get a sense of how different the readings might be. I have a feeling that if this is 7.5 here, we might hit 9 with the Herc probe.
Right. All right, let's come full wide, Aaron.
So talking about octopus intelligence, they have been known to learn how to screw the cap off of a glass jar and get to what's inside.
So we should leave a glass jar of peanuts down here. And see what
Right,
happens.
exactly. Or maybe anchovy.
Anchovies,
Yeah. Do you like peanuts?
They have allergies.
Glass jar of crab.
Yeah, allergies.
Buddy, you were just making this whole looking at the end of the arm difficult.
It's just a skills test, Greg. You didn't want to be able to see your wrist.
Oh.
Tough.
Let go.
Yeah, so we've obviously picked up a hitchhiker here.
These are both at the other vent site, or from the other vent site.
Yeah, there's one on the porch there too. Yeah, well, as you can tell, we didn't grab her. She grabbed us.
So maybe her eggs have hatched and she says, it's time for me to do something else with my life. I'm going to see the big wide world.
Well, I think the theory is an interesting one too, that maybe this venting just removes the sediment and clears hard substrate here, and that may be, it's one of our guesses about why all these octopus may be here. Maybe it does or doesn't have to do with the chemical composition, or doesn't have to do about the temperature. It could just be clear substrate and We just brought a lot of square footage of clear substrate down here with that shiny arm.
There's a pretty evident inverse relationship with the oxygen saturation and the temperature.
Yes, there is. Which is why it's so interesting to find these developing embryos here. But maybe this particular population, they can survive with lower oxygen concentrations. Maybe the warmer water speeds up their development. Maybe it is just the bare rock.
Yeah,
Maybe
so tell us a little more about that.
mineral in the water.
Ooh, like that. Tell us more about that, Chad. Why is warm water less oxygen saturated than the colder water?
That's actually a great question. I know a lot of the oxygen is stripped out when it goes underneath the Earth's crust or into the Earth's crust, but I don't know the chemical process for that.
So that's like microbial digestion of that oxygen?
It can be near the end, but that's not the main reason why that oxygen is stripped out. And we are taking some of the water that's been collected with the squeeze samplers, which we'll probably do here as well, is going to be sent to a colleague of ours at the University of, it's Texas A&M, and he's going to be trying to isolate any potential bacterial presence in the water as well. Hello.
All right, Aaron, let's push back in.
So trying the other tool here,
Hold
this is just important to compare our two different sensors or two different tools. Constrain what we know about the site before we leave behind some samplers, or provide, what we call the metadata. Not only when you grab this water sample do you have whatever interesting analysis comes out of the water.
Logger back up.
Raj.
But you want to know more about the whole site so that can better inform the results of your analysis. So.
Oh no, I didn't want to do that. I just want this to come forward.
Trying to paint the picture here.
Just minimize tension.
Minimize this one. Then we're going to reset the max temp here.
Yeah. We are on our max temp, so it's going to keep just...
Something's wrong here. I can't quite figure it out. I'm going to switch that.
Mm-hmm.
You got a slow climb.
Yeah. Aaron, let's push in more. Hold that.
Are you happy with that placement, or do you want me to brighten up?
I think I'm good for the moment with that placement.
Okay.
If we hit 9, then we're good, and if we don't, then we have to move again.
So Aaron, some people are interested in what the kind of green looking stuff on the rocks is. Could you talk a little bit about just kind of the light pool of the ROVs? Like, sort of towards the edges, the sediment looks like it sort of changes color. I believe that is not actually a rainbow of sediment on the seafloor. It's just...
No, we have a light pool from the ROVs, and as you go out towards the side, the light obviously isn't hitting it quite as much, so you're going to get a change in light, which often makes the color look different.
How much of the light that we see is actually booming down sort of from Argus, and how much is Hercules' light specifically?
Aaron, let's come full wide for a moment. Let me get a Okay, so let's see. All right, let's push back in. Hold that. I'm gonna try and go.
In Between Two Octopus, the movie.
Yeah.
Say it seems pretty solid at 6 for a bit.
Yeah, not too much movement there. Slightly up.
Oop,
Still
moving
going
a
up
rock.
in there.
So again, our team is just watching the temperature readout from this probe, and we're looking to see, you know, we're going for the high score, trying to see what is the highest temperature we can record in this area. But the hot water is sort of seeping out in between the cracks, in between the rocks. So you can imagine just sort of in a jumbledy rubble pile. There are areas where more of the water is coming out or less, and it's mixing with the cold water as soon as it rises away from whatever little crack it's coming out from. So that's why we're kind of stirring around in there, looking for.
So Aaron, we're
Aaron, you want to brighten the probe up? Thank you.
Aaron, that we're reading the temperature on the sonar. So if it's ever appropriate to put that on channel 3, that's where we're getting our. That's where we're all looking.
Okay.
New max temp.
Up to you.
Woohoo, new
Thanks,
record.
Randy.
Wow.
8.1.
Sorry, what was that, Aaron?
I said thanks, Rennie.
Yeah, it all mixes so quickly. I mean, like when we're doing some of the hydrothermal vents and you're in the 300s coming right out of the vent, even at massive flows, by the time it hits the light bar, you're at 5 to 10 degrees.
Yeah, it's so interesting talking about flying in those spaces, because you're like, you really need to not back up into one or fly too close over one, and yet you can sit just to the side of it and you're totally safe.
So we just matched the highest temperature ever recorded here at the spa, 9.7.
Go team.
And
9.8.
Yeah, new record.
And then the overall high score is at the vent at 10.4, but that's such a concentrated flow.
Challenge accepted.
Oh, we're coming for
But
it.
challenge
Yeah.
accepted, yes.
Great to 12, a little bit competitive. So, yeah.
9.83. Seem pretty steady there.
Does this depend on how far we
9.84.
put the probe in?
Yeah, it is. That's why it's kind of a guessing game.
Yeah.
9.9.
Whoa.
Can we get an even 10? Do we hear 10? 10.1?
I see 9.9, 9.9, 9.9, 9.9. Hi to everybody who's tuned in with us.
9.91.
This is so cool to be able to share this with your viewers all over the world right now, following along with us and sending in questions. So we will get to as many of those as we can.
I think that was just like a little wave of something because it's disappeared. We're at 9.85.
Okay.
I do want to refer people, if you are curious how we all got jobs like this, check out the team tab on Nautilus Live. There are brand new career pages there where you can click on the seat, you know, that any of us are in the control room.
9.9 seems like our highest. We're starting to drop down. And we haven't moved, so that just gives you an idea of the different flow.
For sure. Well, I would say this is a prime candidate spot to leave one logger and to try and fire a couple squeezers.
Sounds good. All right, so we'll do the squeezers and then the logger?
Sure, yeah, so the logger doesn't get in the way.
Yeah. Okay, full wide.
Dane, do you want to turn on your pump because we're going to be here for a little while? Or is...
Yeah, let's do that.
Okay. As an official sample?
As an official sample.
Okay.
All right, let me get, actually Paul can get you set up.
Okay.
You can do it on yours. I mean,
So
you can use a touch screen.
And Paul will need a
I don't
countdown.
have the PCB
Oh, he
button.
wants a countdown. Hang
Oh.
on. Okay, you ready for your countdown?
Big fish.
You ready, Jamie?
Not quite.
Okay.
All right, so we're just gonna get set.
Oh, yours hasn't been rebooted.
Chad, what's that fish in the upper left?
Okay, I'm ready.
Okay.
That's a cuskeel.
Countdown to pump. When you're ready.
Okay. 5, 4, 3, 2, 1.
1549.
Thank you.
UTC, time start.
Did I do a full 180 with this? I think I'm good.
No, I think you grabbed and went to the right, if I'm not mistaken.
Yeah, you can look at the shimmer and the exhaust from the sampling pump.
Clockwise.
Yeah.
That's incredible.
Oh.
Perhaps.
So if you're watching on the quad channel, that's in
Maybe
the
I'm
lower
wrong.
left. You can actually see hot water exiting the pump, which is
Or it may be cool water exiting the pump into the warm water. So it's kind of the reverse of what the shimmer is we see coming
Let's
out of
get
the
on
vents.
porch light.
true.
Right, because it's taking water from above the ROV,
Yep.
so
Pulling
that's
it
definitely
down.
going to be cold
Oh,
bottom
off?
water.
Oh, it's on. All right. Let's then just do that.
There.
There's so many good questions coming in that I don't think we have answers for yet. We are just investigating this. The question of are the greatest number of octopus around the warmest vents?
Wow.
Not necessarily. We are finding octopus in all the areas of venting, but one of the objectives for later in this dive is to fly across this whole feature. When we were here a year ago, we didn't get a chance to extensively view the area due to some weather conditions and some just equipment issues that meant we needed to recover, and then we didn't get back into the water in this site. So we have a lot of our just pure exploration. How many vents are there? Did we just happen to get lucky and land right on them the first time? And in that case, we could then say, like, which vents are hotter or colder based on these measurements. But we just have a lot of the basics. How many octopus are out there? How far did these different vents and seeps stretch along this feature? So stay tuned. This is a long dive, and we've got a ton more to, investigate to start getting to some of these questions.
Do we want to try to put the funnel on top of that?
Yeah, in fact, the previous shift, Amanda just called in and said that was problematic last time because of the topography, but she said they believe,
give it a go?
Yeah, give it a go here.
All right.
Will we use the Magnum arm for that?
No, we'll put it out with this and set it down and then go out with it. The Magnum arm won't reach the spot that we're sampling at.
Got it.
All right, you want to go ahead and bump out the tool tray?
Yeah.
Just about a couple bumps out. Yeah, that should be good.
Yeah, Megan, what you were saying about the question regarding the temperature, if we see more at hotter vents. We don't know, first of all, if it's temperature related, as we were saying, but also if there's a threshold or a Goldilocks temperature that they prefer coming out of these vents. I was thinking about, I was drifting for a moment, and I was thinking, I was just in New Zealand, and in the Coromandel Peninsula on the North Island, there's a hot water beach. And at low tide, there's some geothermal activity under the sand, and you walk out, it was winter, so you walk out there barefoot, it's kind of cold, and then you're feeling around for hot spots. And then you dig, and then the ocean water, the seawater fills in, and you have your own little personal hot tub. But the spot that I initially chose was scaldingly hot. So there was all these people surrounding it, and I was in the center thinking I had, made a great discovery, but it turns out
Bad
too
choice.
hot.
Oh no.
So could be the same for some of these octopus.
Yeah, I've had that same experience in Idaho. There are a lot of like mountain rivers that pass by hot springs, geothermal hot springs, and so you kind of like move a few river rocks to divert just enough like super icy cold water in with the super molten water and try and get like a reasonable temperature in the middle. But it can, maybe I'm just,
All right,
I
Aaron,
am the
let's switch back in.
Goldilocks threshold that's pretty narrow, but yeah, it's pretty quick to be freezing or burning.
So just kind of place this on top of those two?
Are you not talking, Chad?
Thanks. Yeah, this is going to be a little challenging here. I know there was a little bit of space above those two, but I don't know if it's too vertical or sloped for it to rest, as opposed to we don't really want to disturb these two mothers too much.
No octopuses were disturbed in the making of this movie.
It's a pretty cool idea, this funnel here. Just to have enough surface area to channel into and then be plugged by the rubber opening. Put a temp probe in there, that kind of...
Yeah, and this is just a PVC cap and some dive weights and a little pipe.
Science is very expensive and very cheap.
Yeah, it is.
Yeah, the means to get this PVC cap down there is...
Yeah, exactly. I'm going to shake this Yankee Candle.
Right, yeah.
Yeah.
I don't think that's going to stay when I let it go.
No, there's nothing on the right side supporting it.
No, there's a little rock behind it, but if I go back any farther to get more on that rock, I'm pushing the other octopus. So this is a
precarious
call by science.
Yes.
I can go ahead and give it a drop.
I'd say go. I don't think it's going to terribly harm them with a small drop because we can pick it up right away if it tumbles sideways. So go ahead and give it your best effort there. I'd say that's pretty decent.
That's great.
Oh, nice catch, Rock.
Yeah, beautiful.
Way to go, Greg.
That's got that's good. It's not disturbing the others too much.
Okay, Herc. So 3 squeezes have already been done.
Right.
It's a little hard to tell. I think in the front row there's 2. One's fallen over on top of the other. Is that correct? There's still 2 in the front row.
So they've used the 3 that you can see the tops of. Those have not been used.
I see. The ones that are more fallen over.
Right. If the pin is extended, it's been fired.
Okay, got it. So, yeah, the next one, if you're going sequentially back, should be number 9 that's available.
Okay. Yep. 9-6.
Yeah, 9-6.
So, Chad, one more time, what are these squeeze samplers going to be analyzed for? What are we going to learn from them?
Most of it's going to be used for water chemistry, what we were talking about a little bit ago, to understand how the seawater has changed as it travels underneath the sediment and in the top layer of the Earth's crust. So we just have a better idea of what the chemical composition is coming out and bathing these eggs in, and just to help understand the hydrological processes. But also a portion of it is going to be used to do some eDNA work on bacterial presence. There is a possibility there's low levels of bacterial communities here. I think right behind
the arm we have a
crawler.
There is a moving one, yeah.
Oh, there is one, yeah. It's possible this is a male.
Right.
But again, that's on the right side. He's hiding it, so.
Yeah.
But we have seen a couple of females crawling around too, and we don't know if those are females that are looking for a mate, if they're looking for a place to lay their eggs, or they're just cruising through.
Sorry to interrupt your last comment there, Chad.
Oh, no problems. No worries.
Actually, there's 2 potential crawlers there.
Yeah.
Yeah, after we do the water samples, we should check those 2 out if one hasn't gone too far.
So then when you come back again, you will just look for these things visually and
Yeah, this is a fairly large area here, so finding it is relatively easy. And then, yeah, visually, we'll just be able to see them, see the
All right,
these,
Paul, if
And
you'll
the black
follow me
ball
in.
at the top is just to grasp? Yep.
I believe so.
The vent? Or the funnel?
Yes.
Yeah, they're hockey pucks.
Hockey pucks.
Yeah, we were taken over by Canadians.
Yeah. But they are inexpensive and easy for the jaws to grab.
That's great.
And you can just machine it, you can drill them. Oh, our roving octopus is going to come into view. It doesn't want to wait for us.
My time is now.
Yeah, I'm a star.
And Chad, for those of those who have never heard of eDNA, can you explain that a little bit?
Thanks. Yeah, so E stands for environmental. So it's environmental DNA, and it's amazing because when I was in graduate school, we were still running gels and looking at different clades of organisms. It was, sequencing had just come out, and it was extremely expensive 20 years ago. And sequencing is basically looking at a sample and looking at the actual DNA pair patterns, the four chemicals that are there. And so every species has a unique assemblage of these letter codes. And it's a much better way to kind of identify species. And so now what we can do is we can sample the water around an area where animals either exist or have even just passed through, and they'll slough off things like skin cells, scales, excrement, fluids. That all has their DNA in there. And so we can look at an assemblage of what exists in that water depending on the primer we choose, if we want to look for a specific set of invertebrates or octopus or bacteria in this case. And then we amplify that DNA, and then we can sequence it, and then that tells us what species exists in the water. So they're using eDNA everywhere now, rivers and streams for invasive species of carp, for whales and big marine mammals that pass through. We don't have them tagged, we don't know where they are, but then we can get their presence in terms of their DNA in the water. So it's really an amazing field of science that's opened up in the last 10 years or so. And we'll be doing that too later in our second dive. We'll be collecting some water for eDNA around corals and sponges to identify them as well.
All right, tons of questions here. We think these are Octopus robustus. We are, just learning about this environment. There've only been a handful of visits here after it was discovered last year. So all of these samples that we're taking are going to help us better understand this environment and answer some of the more complex questions like,
all right, Paul, follow me.
Is the chemistry helping the octopus? Is the temperature helping the octopus? What are the range of temperatures over time?
Okay.
Does it change seasonally,
Just
daily?
to put that in there.
Is it increasing over time? Is it decreasing over time? We have all kinds of different questions just about the processes that are happening here. So the vents are not super hot. As we were saying, hot water, we're talking about 50 degrees Fahrenheit compared to about 30 degrees Fahrenheit. Most of the rest of the water is around here. So it is warm.
All right, Aaron, let's push in.
As we call it the spa, it is definitely warm if you've been spending your whole life in 30 degree water, but it's not hot like denaturing proteins or cooking things like you think about boiling water, like a major hydrothermal vent system.
Let's go ahead and zoom in on bubble a little bit too. Trying to get the very edge of that into the...
Chad, who designed this sampler?
Annie Hartwell would be the right person to ask. I know it's from Jeff Wheat's shop.
Neat.
So, but I don't know the original design, if it came from their shop or if they borrowed schematics or what have you.
Good job.
I know a lot of scientists use squeeze samplers, so I think it's fairly universal.
Jeff.
And where is Jeff Wheat?
Well, he's been associated with Mbari and I want to say University of Fairbanks.
Okay. So scientists from all over
Video,
the
let's push
country participating in this.
Oh yeah,
Oh,
absolutely.
completely.
Yeah, this has brought a lot of scientists together.
Let's try and brighten up that black flap. Thank you.
So that white cap is just acting as a funnel. That there are slits in that black cap. We're going to put the sort of probe end of the sampler in there, where it's actually going to be intaking water. And as you squeeze, sort of like a syringe, but a little bit opposite of a syringe, instead of the syringe pushing out. You can think about as we squeeze this, there's a mechanism that's going to pull in some of this water and capture it in a tube, so that we know we're getting very specifically the water that is rising up through that funnel and out from these vents.
Oh, we're at grip force 9 and we're not going. Okay. So, little wiggle.
Jamie, is this a sample number?
We're gonna have to reposition. All right, science, we're at full grip force and we are not triggering this one.
Full grip force. Okay, we will try the next one.
I can give it a shake over one of the venting areas and see if I can get it to go that way.
Right, it just wouldn't be the concentrate, right?
Are we
Correct.
caught up on the hex lip on the...
I suppose.
You see what I'm saying? On the far left.
Video pushing all the way?
No, it looks like we're under it.
Yeah, we're under.
Yeah.
All right, full wide.
Yeah, I suppose we won't let it go completely to waste. We can just shake it over a venting area, and we get what we get, and we'll try the next sample. We'll just note in the record that this is not, if it does fire, it's not concentrated.
Doesn't seem like a good
Yeah.
grip on it.
Did.
I wonder if the octopus is in there.
Actually, it looks like it's loose in
Yeah.
the claw.
Let's try it again.
Well, let's try that.
So is that a squishy polymer? Like,
Go
should it
back over
squish
it.
really easily?
Video, let's push back in. We're gonna try something. Keep going in. Okay.
There it is.
Oh,
Yes, got
yes.
it.
Good job, guys.
And we'll have to reposition that. All
Sure.
right, full wide.
Jamie, does that have a sample number?
Thanks,
yeah,
Dan.
it does 006.
006.
You may not need to reposition it if you can hit it at an angle, I guess, but leave that up to you.
Yeah, that's great. So you saw that it squeezed, pulled that syringe, sort of, there, that.
So this
the
is
two
squeeze
plates
sample.
back.
Does it have a number associated with it?
It should be number 9.
Should I write that or just forgo?
It is number
I mean,
9.
you don't normally.
It is number 9?
You don't
It's
normally write
Labeled
the
number
numbers
9,
of
yeah.
stuff.
Yeah, I did.
I know the number based on its position.
I see.
Okay.
Yeah.
Just in case.
Yeah, we can't see it from this angle,
we're
but
doing
I know
another
which
squeeze
one
sample.
it should be.
Okay.
Right, so we're gonna try that again. Always take replicates, so you have a couple to compare against each other from the same site, and then also make your comparisons against other sites there.
That octopus is
So obviously,
should move.
it's important to keep track of the numbers so that you know
You're back
when
on
you
grip
get
force
back,
1?
Yeah, no, not yet.
they're paired
I had to get in
correctly.
position, make sure I was happy.
Yeah, absolutely. So
This
Jamie,
is grip force 1. You ready? And close.
So Jamie in the back is keeping track of all our observations in our event log software, so you can log anything from, just observations of sea life on the seafloor, changes in habitat, makeup,
Can I look up a little bit?
geologic
I'll make sure my
features.
other claws are on there.
They're also logging all the
Oh,
samples.
it's right behind the bar. That's where it's supposed to be.
Turning
And
on the pump.
Rennie is also
That means
logging
it's
an
good.
event log, the navigation base, so what locations we are sampling at. When I'm logging video highlights, it's logged in the same system, and all of that is time-coded and stamped to the video so that it all is all correlated. Every data set we have can be matched up exactly with the oxygen concentration, temperature, navigation, depth, CTD measurements of the conductivity and the temperature of the water that the ROV is in, and all of that comes together in our data. So scientists later, whether it's the sanctuary team or another scientist
Is it
interested
possible
in
to
this
rotate
site.
this one to see if there's a number on the backside? You could even flip it upside down or something.
There's a number. It's towards the octopus hangar.
Oh, it is. Okay. 4. Okay, so sample number 4. And that corresponds with the
And
position?
that corresponds, so we should be good.
Okay, good.
Good.
That's what I wanted to do, is kind of cross-reference that.
Is this funnel still in an okay position to be sampling in there?
Yeah.
Yeah, I think
confirmed
so.
that already.
You can
Where you
probably
been, Ryan?
use the squeeze sample to move the funnel a little bit if you need to.
Yeah, but I can only really push, right? So I can't
Yeah.
pull it back up to the top where it was. All right, Aaron, follow me in.
Yeah, so this sensor is just sucking up a little bit of water, of this water that's just flowing through. So with the seeping fluids, we got that max reading of 9.9. Place the funnel,
well lined up.
and then are taking a water sample right from that area.
Nice.
There it goes. You can see the pin extrude
Yeah, the pin
out the
came
right
up.
side,
It didn't, yeah.
and the plunger move on the left side if you're watching
Nav, that's
closely.
007.
Great.
Full line.
Roger.
So we've had a pretty good success at this sample. Right, I was told 60% of these are expected to fire.
Yeah, I'm really impressed.
Let's see, where am I going to put this one?
Looks like my closet hole or something. Just throw it in there.
Yeah, I
Yeah,
definitely
I
have a
might
squeeze sampler
reposition.
addiction.
Squeezy toys?
Yeah. Closet's filled with them.
And so if you're new to the broadcast, welcome. I just love this question. Somebody said, what is up with all these octopus? I just logged on. So welcome to the Octopus Spa. We are on a little flank feature of Davidson Seamount in the Monterey Bay National Marine Sanctuary. This is a site we discovered last year. Not expecting it. It's certainly a brand new discovery, both that there were venting fluids here and that there were so many octopus in this area of venting fluid. So we are back to answer some more questions.
And last squeezy sample here?
Yes.
Sure.
Let's do the last one and check the number and collect.
I can
Yeah.
see the number on bubble is 3.
Yeah.
Okay, see on a bubble. Sample 3.
And that matches, so we're good.
So we are back here to learn more. We think this is Octopus robustus. We do not know how long they brood eggs for, but this is a behavior that is known, that they lay their eggs brood over them, meaning protect them, go into that sort of upside down bowling ball position that we see them in, and they are oxygenating those eggs and keeping them protected.
Really slow.
We're not sure how long they